In this video, we are going to see how ATP is created in cellular respiration. Now, take note, ATP has also been created during glycolysis, which is technically an independent process from cellular respiration, and also from the Krebs cycle or the citric acid cycle. But all of those creation or production of ATP are known as substrate level phosphorylation. So this time around, we are going to study closely how ATP is made in a process called oxidative phosphorylation. So by now, we should be familiar that pyruvate from glycolysis enters the mitochondrion, and the electrons of which are stripped away from the molecule and transferred to the electron carriers NADH and FADH2, which transfer their electrons along an electron transport chain composed of protein complexes in the inner mitochondrial membrane. And this transfer of electrons causes the formation of proton gradients, wherein the concentration of protons or hydrogen ions in the intermembrane space is increased, while the concentration of hydrogen ions in the mitochondrial matrix is kept to a minimum. So in this second step, after that electron transport chain, this difference in the concentration of protons will be used to drive the machinery that creates ATP in a process called oxidative phosphorylation. So notice in this illustration of the mitochondrion that there are actually structures, as you can see here, okay, that look like studs, okay? They look like grains attached to the cristae of the mitochondrion. And they are actually ATP synthase. Okay, so they are enzymes, they are protein complexes, and they sort of look like this one. So this is a three-dimensional image of the ATP synthase attached to the cristae. And they are the machinery that are used to make ATP in oxidative phosphorylation. So this is how one would look like. So this is from a bacterium. And you can see here it is embedded. It is a transmembrane protein complex. It's embedded in the inner mitochondrial membrane. And on this side, we have the matrix side of the mitochondrion, where the hydrogen ion concentration has become really low compared to the hydrogen ion concentration of the intermembrane space. Take note, this is a result of the work of the electron transport chain in our previous video. So, if you can remember how the sodium-potassium ATPase works, that particular ATPase actually uses ATP to pump sodium out of the cell against its concentration gradient, and potassium into the cell also against its concentration gradient. ATP synthase works in a similar way, but instead of using ATP to pump ions against their concentration gradients, it uses the movement of ions through it to make ATP. Okay, so this is a ribbon model of the complex. So it's made up of several protein polypeptides. In fact, it's a factory, a giant factory. Here is a view of the ATP synthase from the intermembrane space. So say you view the the complex from here, from the bottom, you look at it this way, you would see this ring-like structure here colored in green, okay, would be this. Okay, and here is a passageway for the hydrogen ions. Okay, and this is also where the shaft is for this rotating portion of the ATP synthase is this center portion. So we can see here the different subunits. We have the shaft is made up of the gamma subunit, and then we have the beta subunits and alpha subunits that are arranged in alternate surrounding the gamma shaft. Okay, sort of like this one is the axle, and then the wheel is made up of two types of subunits, alpha, beta, and then alpha and beta in alternate. So as we can see here. So notice also that the gamma shaft here in the center is sort of asymmetrical. Okay, it's not perfectly standing up. And that is really by design. Okay, so it's asymmetrical. And here we can see the alternate beta, and then alpha, and then beta, and then alpha subunit. So when this ATP synthase works, it's actually this shaft, the gamma shaft, that rotates, not the whole alpha and beta portions. Okay, so here is the sequence for the binding of the ADP and P and the creation of ATP in the process. So the alpha and beta subunits would actually have an open, loose, and tight conformation in cycle. Okay? So during the open conformation, the ADP and the P can all attach to the alpha and beta subunits. And during the loose conformation, they are sort of attached in place, the ADP and P. And during the tight conformation, this is a time when they react to form ATP here. And then there will be the open conformation again, so the resulting ATP will be released out of the alpha and beta subunits. So we can see here how the rotation of the gamma subunit here in the center dictates the state of the alpha or the beta subunit, whether they be open or closed. Okay? So for example, in this sort of sharp portion of the gamma subunit, wherever this is facing, the conformation of the beta or alpha subunit here is open. Okay? So meaning to say the ADP and the P can bind in. Okay? And take note here, the open conformation has moved to the green colored subunit. That's because the sharp portion of the gamma subunit, which is constantly rotating, is now facing here. Okay? And this portion, the blue one, is now in the loose conformation. And as the gamma subunit continues turning, eventually this will be in tight conformation, and the ADP and the P can react together to form ATP. And we see here in this last portion, the pointed portion of the gamma subunit is now in this red subunit. So meaning to say this is the one that is in open. Okay? And the ATP here may get released from the alpha or the beta subunits. So how do scientists know that this happens, that the ATP synthase indeed sort of rotates? So they made an experiment. They sort of attached the ATP synthase with all of the subunits, gamma, alpha, and beta, onto a cover slip, okay, using a His tag. And then they provided this with a mixture of ATP. Okay, so the inverted and attached ATP synthase actually converted the ATP to ADP and P. a reverse. And the rotation of the gamma subunit as viewed from the membrane side is counterclockwise. Okay? However, in the real mitochondrion, when this is still working in the living mitochondrion, okay, this rotation will actually produce ATP from ADP and P, sort of the opposite of what's happening in the experiment. And the rotation from the membrane side is in a clockwise direction. So you think of the ATP synthase like a generator. Okay? When a generator rotates in a certain direction, it produces electricity. But when it rotates in the opposite direction, it sort of uses electricity so that it becomes a motor. So we have here sort of a generator motor engineering feat found in the mitochondrion known as the ATP synthase. Okay, so that is how ATP is made. And here we can see a summary of how the ATP synthase here is actually located also on the inner membrane together with the electron transport chain complexes that we see here. Okay, so the ATP synthase is not just found in the mitochondrion, it's also found in the chloroplast and even in prokaryotic cells. And it is used by these structures to create ATP.